Bismillah Rahman Rahim. We start the questions on chapter 5 and enzymes. I've already uploaded 2 videos on these chapter. Now I'm going to show you how to handle the questions on this chapter. Now the first question is enzymes are biological catalyst. Define the term catalyst. Now here, what are you going to write? I'm sure, I look at check papers and I see somebody would have written they're protein in nature. Then somebody would have said they're found in living organisms, because biological, and they're concentrating on the word biological, so found in living organisms, and then they said speed up reactions. But you see, when you're reading a question, remember you're not reading the question properly. It says enzymes are biological catalysts. Define the term catalyst. So a catalyst is only something which is going to speed up reactions and is not going to be used up in it. So they haven't asked you the word biological to explain the word biological, but if they said, okay, enzymes are biological catalysts, explain what does it mean? And probably would be a 3-mark question, but here it's only a 2-mark question. Now, if you've written protein and living organism, well, you don't get any marks for that, because the mark scheme is very clear. The mark scheme says that all you had to give me was that it speeds up reactions and it's not used up in the reaction. Oh, you have to be very careful in how you word your answer. And this is a very important skill, is to develop the right biological English. So enzymes are biological catalysts. Define the term catalyst. Now, the only 2-mark scheme points which were allowed, this is a 2-mark question. The only 2-mark scheme points were allowed. It increases the rate of a chemical reaction. Chemical is in brackets, so if you didn't write that, it doesn't matter. And is not changed by the reaction. Again, the things that I've written in bracket, it should imply that, but you don't need to have written that. So everybody could get 2 out of 2. It's a very straight question. But then if you had written about proteins and amino acids and other things and the fact that they occur in living organisms, and you hadn't written these 2 points. Remember, if you had written those 2 points and you had written these 2 points as well, then I would have given you the 2 marks. But if you had written the points that they are proteins and they are found in living organisms, then I wouldn't have given you any marks. But if you had given me 4 points and 2 of them were correct, but I would give you your 2 marks. The next question is, pectinase is an enzyme. The box on the left shows the beginning of a sentence. The boxes on the right show some endings of sentences. Draw 3 lines from the word enzymes to make 3 correct sentences. Now pause the video here before you see the answer and try to do it yourself. So enzymes are living organisms. No, enzymes are not living organisms, they are molecules. So I would not put a line there. Enzymes are proteins. Yes, I'd put a line there. Then enzymes can only be used once. No, that's wrong. They cannot only be used, they keep on being used again and again. Then have a complementary shape to their substrate. Yes, that is correct. I put a line here. Increase the rate of chemical reactions. Yes, I'd put a line here. The last point is, in the stomach are most active at pH 8. Now that's totally wrong, because the pH in the stomach is very acidic. It's 2 or 3. So this is wrong as well. So 3 lines, 3 marks. If you get those 3 lines right, you got your 3 marks. Now coming to question number 9. The cut surface of an apple usually turns brown when exposed to air. Well, you've always seen when you're eating an apple, and if you leave a piece of that apple after some time, it turns brown. This is caused by enzymes. Now, the brown is because the enzymes are working. Now, 3 slices of apple were given different treatments and were then exposed to air for 30 minutes. The table shows the treatments and the appearance of the apple slices. Now, apple slice A, the pH was 6, the temperature was 20 degrees Celsius, the appearance of the apple at 0 minutes was white and it turned brown. So here we see the enzyme are working. But in B, if you see B, the pH was 2, the temperature was the same, the appearance in the beginning was white, but it remained white, so enzyme is not working. You see, when you read the question, you've got to understand how do you follow the biological line of thinking. So in C, when you look at C, the pH was 6, the temperature was 4 degrees Celsius. The temperature here is in degrees Celsius. The appearance in the beginning was white, but then it remained white, so again enzyme is not working. Now suggest Why slice B and slice C did not turn brown after 30 minutes? Well, if you think of it, we're talking of temperature and pH. That is your syllabus point. So in this, the pH was 2, so this did not work. Here, the temperature was 4, so it did not work. But it worked at 6 pH and 20 temperature. So that means this would be the optimum pH and the optimum temperature for this enzyme. So slice B and slice C remained white, means enzymes did not work. What are the different ways that you could express this? You could have said pH is too low because the pH was 2. Or you could have said not the optimum pH. When I put a slash here, it means you could have used this was the alternative possible answers. So not the optimum pH, or you could have said enzyme stops working at low pH. So this was the English that we were expecting you to write, because you must word it correctly. If you just said it was not the right, the pH and the temperature was not correct. Well, what was not correct? What was wrong? It was not correct. What do you mean it was not correct? Was it low? Was it high? Was there something else wrong? So you have to be very correct in answering this question. For slice C, you could have said temperature too low. 4 degrees was too low a temperature. Or you could have said it is not the optimum temperature, because the optimum temperature at which the enzyme worked was 20 degrees Celsius, because it turned brown in that. So 20 is the optimum. This was 4 degrees, so it was much lesser than the optimum. Then coming to question number 13, the reactions of chemical digestion are catalyzed by enzymes. The diagram shows the stages of an enzyme catalyzed reaction. So we have an enzyme which they've labeled. Then we have A, and then we have B, and then we have C, and then we have D. State the names A to D in the diagram. Now what is A? A, of course, is an enzyme. Or A wasn't, sorry, A is not an enzyme. A is the substrate. So A is the substrate. What is B? B is this area which they have labeled. So B is the active site. And C is the enzyme substrate complex, because the enzyme and the substrate have binded to it. So it's enzyme substrate complex. And then what is D? D is the products. So all that they'd asked you was state the names A to D in the diagram. So you don't have to give me any explanations. You just have to write this: substrate, active site, enzyme substrate complex, and products. Now in this question, the first diagram represents a chemical reaction, and you've seen the reactants and you've seen the product. Now it says which diagram below represents the enzyme for this reaction. Now I have colored this and shown you how this would have been formed, and because this would fit here, so the answer is D, because you see this is where it would fit. This whole thing would be something like this, which is the whole thing which is going to be formed. So the answer was D. This is an MCQ. Then coming to question number 17. Now this is a very difficult question. In an investigation to measure the effect of pH on enzyme activity, a protease enzyme, read the question very carefully, a protease enzyme was mixed with solutions of different pH values. So we had the basic enzyme was protease, but we took different pH values. The mixtures were placed in holes cut in the center of dishes of gelatin. And gelatin is a protein which can be stained with a colored dye. So we took gelatin and we stained it with a colored dye. When the protease digests the gelatin, a colorless zone forms. So we have this gelatin, which is all a certain color with a certain dye, and then we put the protease in the center of it. Now the protease, when it is going to digest it, it gives a colorless zone around it. So you understand, the colorless zone means the enzyme is moving out of it, digesting it, and the protein is being converted to amino acids, and amino acids are soluble. So they give you this colorless zone. So the larger the colorless zone, the more the enzyme is working. So first of all, there's a colorless zone which is smaller. The colorless zone is even less. So that means the enzyme is the size of the colorless zone is deciding how well the enzyme is working. If the colorless zone is larger, it means more amino acids have been formed and the digestion is better. So gelatin is a protein which is stained with a colored dye. When the protease digests the gelatin, a colorless zone forms. The diagram below shows a dish at the start of the experiment. Now here is the picture. Stained gelatin, a protein which I have stained black, right? A hole containing the mixture. This is the hole containing the mixture of solution and protease. Containing mixture of solution and protease. And this is the dish at the start. The diagram below shows the dishes after a period of 24 hours. Now look at the colorless zone. At pH 2, gelatin, colorless zone. Then pH 4, then pH 6, then pH 8, and then pH 10. So you can see the size of the colorless zone. This is the important thing which you've got to understand, the size of the colorless zone. I'm going to give the size of the colorless zone a blue color. So just for you all to realize. So this is the colorless zone here. This is now a little bigger, the colorless zone. Here it's even a little more bigger, the colorless zone. And here it is even bigger, the colorless zone. So as we are changing the pH, at 10 it was not working very well, at 8 a little better, 6 better, 4 even better, and 2 has the largest colorless zone. So that means this protease is working best at pH 2. Now using the diagram that shows the dishes after a period of 24 hours, state the optimum best pH of the activity of this protease. Naturally, it is pH 2. Why? Because that has the largest. So this is the pH 2. Then suggest the region of the alimentary canal. Naturally, that is the stomach, because where else does such a low pH exist? Nowhere else in the body. So stomach, then complete the equation to explain how the protease caused the colorless zone. So basically, what have you to say? You've got to say that the protein in the gelatin, I'm giving this in bracket, is converted to amino acids. Now coming to another interesting question, researchers in Michigan investigated the rate of photosynthesis in leaves of big tooth aspen trees, a Populus grandidenta, by placing some of the growing leaves inside transparent boxes. The researchers measured the uptake of carbon dioxide by the leaves over a range of temperatures, from 10 to 40 degrees Celsius. They carried out their measurements at two different concentrations of carbon dioxide. H was at 325 parts per million carbon dioxide. So H was at 325 parts per million and J was at 1935 parts per million. Carbon dioxide, which is a very high concentration. They also told you that. So this was a high concentration of carbon dioxide. And then the results are shown in the graph below. Now you can see here the temperature is being studied. So the carbon dioxide is more in J and it is less in H. So this is H and this is J. Now explain by the rate of photosynthesis in the leaves in batch J increase with increase in temperature from 15 to 35 degrees Celsius, and number 2, decrease at temperature above 35 degrees Celsius. You must read the whole question before you start answering it, because sometimes you write it in the wrong way. So if you wrote something here and that was in the mark scheme here, well, you won't get any marks here, and you can't be writing the same thing in both the parts of the question. So always read the whole question before you start answering it. Now you can see this is a very varied question, because only for 2 marks there's 6 mark scheme points. Number 1, Temperature is a limiting factor. Number two, increasing temperature increases kinetic energy or increases the rate of diffusion or increases the movement of molecules. Number three, more effective collisions between substrate and enzymes. Number four, to speed up chemical reactions. Increase in temperature. Keep it in mind. Number five, stomata open wider. Number six, more CO2 entering the leaf. So out of these 6 points, you should have given me any 2 points to get your 2 out of 2 in this question. So I always say write more so that you can then be smart. That's called smart work. That you can be sure that you've written at least 4 points and 2 will be correct, and so you'll get the 2 out of 2 in this question. Now the second part says, how will you explain this decreases at temperature above 35 degrees Celsius? Why does the rate decrease? And you remember this graph, why does the rate decrease? So we've got to be thinking of why does the decreases at temperature above 35 degrees Celsius. They haven't given us those here, but why would it decrease after 35 degrees Celsius? Basically, what is the reason? You know, enzymes are denatured. Enzymes are no longer active. Or you could have said stomata close, therefore reduced carbon dioxide entering the leaf. So enzymes are denatured, enzymes no longer active, stomata closed, therefore reduced carbon dioxide entering the leaf. Because higher temperatures causes the stomata to close. This is a way of their survival mechanism. Too much temperature would mean too much water loss. Water vapors would diffuse out, so that would cause wilting of the plant. So this is a safety mechanism of plants, is that when the temperature is too high, then the stomata close. Of course, for that time, photosynthesis will be compromised, but it will save the plant. Now coming to question number 22, one of the components of human milk is the enzyme lysozyme that is present in many body fluids. It's present in our tear fluids as well, and is responsible for breaking down cell walls of bacteria. Define the term enzyme. Now the enzyme, what could you have written? You could have said enzymes are catalyst, biological catalyst, or they're made by cells, got you 1 mark. By saying catalyst speeds up rate, you could have said they're biological molecules, then you would have only got 1 mark. But if you said they're biological catalysts, then you would have got 2 marks for that. In one sentence, you would have got 2 marks. And then they're made up of proteins. So for 2 marks, there are 3 mark scheme points. But any two of these, maybe you just say it's a catalyst and they're made up of proteins, well, then you would get your 2 out of 2. So the word was in a previous question, remember I asked define the term catalyst. Here it is saying define the term enzyme. So when you're talking of enzyme, then we're talking of they are biological catalysts. So this is what was expected from you. This is a difficult question, and I'll try to go as slow as I can. The effect of human lysozyme on two common species of bacteria A and bacteria B. So A, and B. Read the question very carefully. Was investigated at two different pH values. The investigation was set up as shown below. The test tubes were kept at 37 degrees Celsius for 24 hours. So we have what? We have species of bacteria. We have A in tube 1, A in tube 2, and A in tube 3. And we have species B in tube 4. So here we have species B. pH of the medium was 4, 4, 9, and 4. So in pH 1 and 2 and 4, the pH was so, and the 4, so they were all 4. 4 here, 4 here, 4 here. Only in 3 we had a pH of 9. Okay? Now we have fresh lysozymes in tube 1, in tube 3, and tube 4. But we have boiled lysozyme. So we have boiled the enzyme. Lysozyme is an enzyme. They've told you this in the question. So it is boiled in tube 2. So in tube 2 it is boiled. And the enzyme, what does it do? It destroys the cell walls of bacteria. This is what the previous part of the question that we did. We read that it destroys, present in many body fluids, breaking down the cell walls of bacteria. Then after 24 hours, samples were taken from each test tube. Each sample was placed onto nutrient agar in petri dishes. The dishes were incubated at 20 degrees Celsius for a further 24 hours to allow any bacteria to grow. So we grow bacteria on petri dishes. The results are shown below. We took the samples and then we grew them on these samples from test tube 1, 2, 3, and 4. And what do we see? Results after incubation. So no growth of bacteria in tube 1. But growth of bacteria in tube 2 took place, in tube 3 took place, and 4 took place. So growth did take place in tubes 2, 3, and 4. You see, always when you read the question, explain this to yourself. Now, what are they asking you? Now, they are asking you a very simple but a very difficult question. You have to compare 1 and 3, 1 and 4, and 1 and 2. The question was, explain. Explain means you got to give me a lot of bio. Explain the results shown above by comparing the following pairs. Which ones are you to compare? 1 and 3, 1 and 4, and 1 and 2. 1 and 3, 1 and 4, and 1 and 2. Now let's compare 1 and 3. There was no growth in 1, but there was growth in 3. Now why is that? Why is it? Because lysozyme is active in 1, because the pH is 4 and it is the correct pH. And what has happened is that the cell wall is broken down or digested and destroyed in tube 1. So there is no bacterial growth. So there is no bacterial growth in tube 1. So 1 and 3 ka comparison makes sense. 1 and 3, 1, there was no bacterial growth. As you can see here, there's no bacterial growth here. So when we are comparing 1 and 3, we say, okay, why? Because in 1, there's bacterial growth. In 1, there is no bacterial growth. So what has happened? It was the correct pH and the lysozyme was acting. So tube 1 and 3 comparison, I've given it to you in writing. Lysozyme active in tube 1, cell wall of bacteria broken down. So these are 1, 2, 3 separate marking scheme points, but you have to give me any 2 to get your 2 out of 2. Now let's compare tube 1 and 4. Now when you compare tube 1 and tube 4, tube 1 and tube 4, now what you have to understand is that there is bacteria B here and bacteria A here. So what is happening? Now there is bacteria has been destroyed in A, but bacteria has not been destroyed because there is no growth in 1 but there's growth in 4. So what is the reason? The reason is that the lysozyme destroys the bacterial cell wall in the species A, and the lysozyme does not destroy bacteria B, which is in tube 4. So the cell wall is differently made, and the lysozyme cannot destroy it, doesn't have the substrate, it doesn't have the right molecule which is acting as a substrate for the enzyme lysozyme. So it is specific to bacteria A, but bacteria B is resistant. So this is how you would compare 1 and 4. So how would we write it? Lysozyme destroys bacteria A, lysozyme does not destroy bacteria B, and bacteria B is resistant. So 3 points, but you have to give me any 2 of them, not all 3 of them. Now the last part of the question and the last of this video is you have to compare 1 and 2. Now if you compare 1 and 2, what was the difference? What was the main thing which was different? pH was the same in both. Here it was a boiled enzyme. That was the only thing which you had to really decipher. So in test tube 2, because it was a boiled enzyme, so the lysozyme was denatured by the boiling and the lysozyme was not active anymore. The shape of the active site was lost. And tube B is actually, the tube 2 is a control to show that lysozyme is responsible for no growth in the tube. Now how would we write this? Lysozyme denatured by boiling, lysozyme not active, and tube 2 is a control to show that lysozyme is responsible for no growth in tube. You see, and no growth in tube 1. So what was happening is that the lysozyme is an enzyme which destroys bacterial cell walls. So in this situation, the lysozyme was working. So it destroyed the cell wall, so there is no bacterial growth here in tube 1. But in tube 2, there is bacterial growth. So why is there bacterial growth? Because now the lysozyme has been denatured, and the lysozyme which was destroying the cell wall of bacteria is now not working. So the bacteria now are okay and they are growing. So their growth is there and their growth is present. So this is actually a comparison with tube 1 in which lysozyme was working, so the bacteria were destroyed, so there was no bacteria growing in tube 1. But in tube 2 the bacteria is growing because what have we done? Lysozyme is the killer of the bacteria. Lysozyme destroys cell walls of bacteria, so there should be no bacterial growth. But because here we have boiled the enzyme, that is why there is the bacteria now are partying, party time, because they now don't have any deterrent. They don't have anything which is going to destroy their cell wall. So please understand this question. I know this is a very technically difficult question. I've gone a longer in explaining it so that everybody is very clear on it. Thank you very much. Thank you for watching. As I end, I usually say this little prayer for all of you and for myself as well. Oh Lord, give us in this world that which is good, and in the hereafter that which is good, and protect us from the punishment of the fire.